So PCR is a method for rapid production of a very large number of copies of a particular fragment of DNA. So let's say you have already produced the insulin gene by reverse transcription from mRNA. So you've got very few copies. So you want to amplify it. So we use PCR method. So what is PCR method? So you want to make many copies, right? This is called amplification or cloning. Now, PCR method is a cyclic process that involves 3 steps. So after these 3 steps finish, it will repeat again 1, 2, 3. Then after that, you will repeat again 1, 2, 3. That's why it's called a cyclic process. Okay, so the 3 steps will continue to repeat itself. So step number 1 is denaturation. So you got your piece of gene already. Step 1 is to denature this gene, means you want to cut the hydrogen bonds between the complementary bases. So you need high temperature to break the hydrogen bonds to separate these 2 strands. So this is happening at 95 degrees Celsius. You need to memorize it. You need to memorize this procedure. Okay. Next step is annealing. Annealing means to join. And this one happens at 65 degrees Celsius, which is actually lower. So when you got lower temperature, it allows hydrogen bond to form. So what do you want to join to the DNA template? You see this red color region? It's actually a short length of DNA, which is single-stranded, that will form the 5' end. It will form the 5' end of the new DNA strand, right? So this short chain of DNA is called the primer. So we call them the DNA primer. So you can write this as DNA primer. So the annealing process is to add DNA primer to the template strand to form the 5' end of the new DNA strand. Okay, you add it to your template to form the 5' ends of the new DNA strand. So this must occur at 65 degrees Celsius because this temperature is low enough for hydrogen bond to be formed. After this is ready, then the next step is called elongation or extension. So we will use DNA polymerase to catalyze the addition of DNA nucleotides by complementary base pairing. So it will be added to primer, and then it continues to elongate all the way until the end by complementary base pairing. So this process is catalyzed by a DNA polymerase called Taq polymerase. So therefore, from one DNA, after one cycle, 2 DNA will be formed. Then after that, the cycle will repeat itself. So from 2 DNA, it becomes 4. Then repeat again becomes 8. Repeat again becomes 16 and 32 and so on. Okay, so this is called PCR. This is a very fast method to amplify or to clone many, many copies of the DNA. So just now I mentioned about the primer, right? So if you're asked to define what is a primer, it is a short length of single-stranded DNA because it's short. In bracket means no need to memorize. And it's chemically synthesized one. It serves as a starting point for DNA synthesis because DNA polymerase can only add new nucleotide to existing strand of DNA. So it cannot add nucleotide to nucleotide. It must add nucleotide to a short DNA strand. So therefore, we must add primer first. So it contains base sequence complementary to the start of the DNA template strands. So if the primer is 5' end, that means the DNA templates must both be 3' end, right? This side also 3' end, right? So the template is 3' end. Now, just now I said Taq polymerase. Now, Taq polymerase is actually DNA polymerase, but it's called Taq in italic because this polymerase comes from a special bacteria called Thermus aquaticus. So the word TAQ comes from Thermus, T, aquaticus, AQ. Thermus means something to do with thermo. Thermo means hot. Aquaticus has to do with water. So this bacteria naturally live in hot spring in the Yellowstone Park in US. So they actually discovered this bacteria there. And because it lives in hot spring, its enzyme optimum temperature must be very high. So therefore, the scientists have decided to use this enzyme in the PCR method so that they can raise the temperature to 72 degrees Celsius to increase the speed of this process, right? You increase temperature, you increase the rate of reaction, to increase the speed of process without denaturing this enzyme. So they don't have to keep on changing the enzyme because the enzyme will continuously to be active. So that's why they use this Taq polymerase, okay? So recap, what are the raw ingredients that you need for PCR that you put into this tube here? Can you see this tiny little tube that you put into the machine to run PCR? So the tube must contain, of course, number one is your DNA, the gene that you want to replicate. Then you must have got the primer. Then you must have the Taq polymerase. And then these nucleotides are called the DNTPs. Please remember to write DNTPs, deoxynucleoside triphosphate for DNA, is DNA nucleotides. Okay, and of course there's a buffer solution because you need to maintain the pH at optimum pH. So therefore you got buffer solution. So these are all the raw materials that they put into this tube, tiny little tube, right? And then place it into the machine. And this tube has got very thin wall to allow very quick change of temperature with regards to the temperature that's needed. So the machine will provide the temperature, and then the temperature will change very quickly because the wall is very thin and the volume of the solution is only 0.05 ml, very little. You can write down if you want to. So let's answer this question. How many molecules of DNA are produced starting from one double-stranded DNA after 8 cycles of PCR? Okay, who can answer this question? Please type your answer. After 8 cycles, how many DNA will you form? So since it's 8 cycles, that means every cycle you double, so the answer should be 2 to the power of 8. So 256, very good. Okay, 256. 2 to the power of 8 is equals to 256. Correct? Okay. Now, next question. Explain why it is impossible to use PCR to increase the number of RNA molecule. Try. So you cannot synthesize RNA molecule because if you want to synthesize RNA molecule, you need to have got free RNA nucleotides, right? Where you don't use free RNA nucleotides, you use free DNA nucleotides. So you cannot synthesize RNA. And then we are using DNA polymerase, the Taq polymerase. We are not using RNA polymerase, so we cannot do that. So that's the reason we cannot. Now, this is a possible application question for this part in the exam one, you know. I may not ask you or tell me all the processes of 3 steps in PCR. I just give you this question. This is called higher order level thinking question for paper 4. Okay, A2 questions are something like this. So you need to think and give the answer one. It's not so direct. How many % of them not so direct? I'll say about 40 to 50% of the questions are not so direct. Or maybe 60 sometimes. Okay, so therefore it's important for you to do passive question and start now. Start now. Don't wait.